Good morning, children. I hope you all are fine. Yesterday, we have studied about what is inheritance, variations, why Mendel selected Pisum sativum plant for his experiments, and then about certain terminologies. Today, let us discuss about inheritance of one gene, or it is called single gene inheritance. In this, the first one is about monohybrid cross. You know very well that mono means single. So, it is the cross between two plants which differ only in one pair of allele or character. Got it? It is a cross between two plants which differ only in one pair of allele or character. That means each character is controlled by a single gene. That gene has two alleles. So it is the cross between two plants that differ in only one character. So it is the cross between individuals having contrasting traits. So what is a monohybrid cross? Cross between two plants that differ in only one pair of allele or a character. So let us come to the next one, how to do this monohybrid cross. So here first he has taken a tall plant, and you know very well, yesterday you have studied tall is a dominant character. He has taken one pea plant which is tall and the other one is of dwarf, and tall is a dominant character, dwarf is a recessive character. So to represent tall, he has used capital T because tall is dominant. So which letter is being used? Capital T is used. And how many times written? Twice. Why twice written? Because organisms are of diploid. So here it is capital T, other one is small t, small t. This is clear. Two plants he has taken. One is a tall plant. Tall is a dominant character. To represent the dominant character, capital T is used. Twice written because organism is of diploid. Now to represent dwarf, small t is written because dwarf is a recessive character. To represent the recessive character, we have to use the small letter of the dominant form. So we are using small t, small t. We are not supposed to use small d, small d. This is clear. Now next one is about the gamete formation. Here tall, homozygous tall. Both the alleles are identical. So gamete is always haploid. So only one T is written and that should be encircled. Clear, it is being encircled. Likewise here only small t is written. And then during fertilization both the gametes fuse with each other and formed the F1 generation or first filial generation. So what is the phenotype? Phenotype is tall. The plant is externally tall. What is a phenotype? The external appearance of an organism. So here the F1 generation plants are tall. So phenotype it is tall. What is the genotype? It is heterozygous tall. Clear, both the alleles are not identical. So it is a heterozygous tall. Now he has taken two F1 plants and that were crossed with each other. Now again here the F1 plant is a heterozygous tall. So it produces two gametes, one with capital T, other one with small t. Another plant is also producing the same one. So when you are crossing this, you will be getting the phenotypic and the genotypic ratio. So here you can see parental generation is represented by the letter P, First you have taken tall plant and then gamete encircled F1 generation heterozygous tall, and two F1 plants were taken here and they were crossed. Can you see now? So first what is being taken? This is one gamete of female parent, another gamete of female parent. This is one gamete of the male parent, another gamete of the male parent. So first what is being done? This T is crossed with this one, capital T, capital T. Second is capital T with small t. Third one, capital T with small t. So always you have to write the dominant form first, followed by the recessive form, then small t, small t. Now if you count how many are tall, 1, 2 and 3 are tall and only one is dwarf. So phenotypic ratio is 3:1. What is the genotypic ratio? One is homozygous tall, whereas these two are of heterozygous tall and only one is dwarf. So the genotypic ratio is 1:2:1. This is clear. For a monohybrid cross, always the phenotypic ratio is 3:1 and the genotypic ratio is of 1:2:1. Now let us pass on to test cross. What is a test cross? It is crossing back of F1 organism with the recessive parent, or the significance. It has a unique significance, that is to know the purity of an individual. What do you mean by purity? Purity means you are having a plant. We are not knowing whether the plant is homozygous tall or heterozygous tall. So the main advantage of test cross is to find out whether the organism is homozygous or heterozygous for a character. So it is the crossing of an unknown genotype with the recessive parent. Crossing an unknown genotype with the recessive parent is called as test cross. The significance is to know whether the plant is homozygous or heterozygous for this cross. So here, for example, here you can see a purple flower. We are not knowing this is a homozygous purple or heterozygous purple. We want to know now. For that reason, now we have taken it. Under which condition we have taken? Under homozygous condition we are taking. Capital, that is both are under homozygous condition. With which one we are crossing? With white flower we are crossing. In the F1 generation, all are of violet. Then the unknown one is homozygous dominant. We have got a purple flower in a pea plant. We are not knowing whether it is homozygous or heterozygous. For that reason, we have crossed this plant. With which one we have crossed? We have crossed it with the one which is producing white flower. So first we have taken under which condition? We have taken it under homozygous condition. And after the cross, what had happened? All the flowers are of violet. That means the unknown organism is homozygous. Second time we have taken it under heterozygous condition. Again we are crossing it with which one? We are crossing it with the white flower. Here we are getting half, 50% flowers are of purple and other 50% are of white. That means after the test cross, if we get 1:1 ratio, the unknown organism is heterozygous. After the test cross, if all the organisms are of same type, then the unknown organism is homozygous. Clear? Test cross is crossing an unknown genotype with the recessive parent. After the test cross, if all are same type, unknown is homozygous. After the test cross, if you get 1:1 ratio, unknown organism is of heterozygous. After this monohybrid, Mendel has formulated two laws of inheritance. First law is called law of dominance. First one is called as law of dominance. According to this law, characters are controlled by a discrete unit called factor. During Mendel's time, he has not used the word gene, rather he used the word factor. Characters are controlled by a discrete unit called factors, and factors always occur in a pair. You know that each gene consists of two alleles, so factors occur in a pair. In a dissimilar pair, one member of the pair dominates the other. So for example, let us take heterozygous tall, capital T, small t. Which one is expressing? Tall. Capital T is expressing. So in a dissimilar pair of factors, one member dominates the other. This is what is called as law of dominance. So law of dominance states characters are controlled by a discrete unit called a factor. Factors always occur in a pair. In a dissimilar pair, the dominant one expresses itself by suppressing the recessive one. So example is of monohybrid cross. Here the phenotypic ratio is of 3:1, genotypic ratio is of 1:2:1. Now the second law is called as law of segregation. That law is called as law of segregation. It is the universally accepted law of inheritance. Mendel has proposed three laws. This is the second law, and it is the universally accepted law of inheritance. It states that the allele pair separate or segregate during gamete formation, and the paired condition is regained during zygote formation. This is clear. You have taken capital T tall, small t as dwarf. But this is called the allelic pair. The allelic pair separate when in the gamete. When you are writing in the gamete, one capital T you have written within a circle. Gametes are haploid. Again the paired condition is regained when in the zygote or in the F1 generation. And it is the universally accepted law. So it states that allele pairs segregate or separate during gamete formation, and the paired condition is regained during the zygote formation or in the F1 generation. Next one is about incomplete dominance. It is called as incomplete dominance. So complete dominance means in complete dominance always the F1 resemble one of the parent, right? Incomplete dominance. Here the dominant allele is not completely masking the effect of recessive allele. Actually dominant allele means it has to mask or it has to suppress the effect of recessive allele, and it has to express itself. But in incomplete dominance, what is happening? The dominant allele fails to mask the recessive allele. As a result, the F1 generation is intermediate between both. In case of complete dominance, the F1 resembles one of the parent. But in incomplete dominance, the dominant allele is not masking the effect of recessive allele. As a result, the F1 generation is intermediate between both the parent. So it is also called semi-dominance or co-dominance. It was first reported by a scientist, Correns. Dominant and recessive were first reported by Mendel, but here incomplete dominance was first reported by a scientist, Correns, and he was noted this one in 2-3 plants. One is of Mirabilis gelapa, other one is called as dog flower or snapdragon or antirrhinum. only these plants show the phenomenon of incomplete dominance, where the dominant allele fails to suppress the recessive allele completely. As a result, the F1 is intermediate between both the parents. But it is not seen in all the plants. It is seen in Mirabilis gelapa, and then in case of dog flower or snapdragon. You see here, this is which one? This is a plant having red color flower. Another plant is having white color flower. One is having red, other one is white. Normally what we will interpret, red is dominant, white is recessive. So when we are crossing these two plants in the F1 generation, we expect which one? Red color. But to our surprise, Coran got which color flower? Pink colored flower. So it is an example for incomplete dominant. A plant having red is crossed with another plant of the same one having white flower. But in the F1 generation, all of pink in color. Now in the F2 generation, so here red is dominant. Red is dominant. So we have used which one? Capital R, capital R. White is recessive. So we use small r, small r. But in the F1 generation, we got capital R, small r, right? But we are expected to be red. But it is which color? In reality, it produced pink colored flower. Now, when two F1 generation plants were crossed, here can you see here capital R, small r, capital R, small r. That time we got one red, two pink and one white. So, both phenotypic and genotypic ratio here is the same. One red, two pink, one white. 1:2, 2:1. The both phenotypic as well as genotypic ratio remains the same in case of incomplete dominance. Go through up to this. If you have any doubt, you can clarify it. Thank you, children.